Hello, everyone. Happy to be with you in the Abromina School. My name is Mohamed Salama. I'm Associate Professor at the Institute of Global Health and Human Ecology in the American University in Cairo, Egypt. I'll be talking about precision medicine and translational neuroscience approaches, especially in the area of neurodegenerative disease. Okay. First, I would love to introduce the concept of translational research. Usually when we start talking about translational research, especially from perspectives of scientists or biomedical scientists, we start to say that translational research means crossing the gap between basic and applied or clinical research. And that's true. However, we forget to mention the second part of the definition. So actually, we have two types of translational research. First is, as I said, crossing the gap between basic research, which means research inside the lab, to clinical application, how to take your idea, your innovative idea into clinical application in the clinics and on patients. However, the second part of translational research is how to convince community to start adopting your solution. So it's not only coming up with a new treatment or new diagnosis, but also how to convince patients and clinicians and the broad category of the community to start adopting the solution and start taking your treatment or using your investigation. With that said, currently we are dividing translational research into T1 and T2. Most of scientists are fond of T1 approach. So we are all the time thinking about how to cross this gap, and that's very important. However, it's equally important and maybe even more important to think about how to approach the community, how approach patients, how approach the broad category community of physicians with T2. And that's why it is important, crucially important, to start thinking of both approaches. As we said, it's not only T1, there should be always T1 followed by T2. Right. And this is a very nice diagram I usually use. It presents the definition of translational research by the word or by the code crossing the valley of death. Why it is valley of death? To understand this concept, actually try to go to PubMed, for example, and type the word neuroprotective agents for Parkinson's disease. Okay, you'll find thousands, maybe hundreds of thousands of papers talking about trying different neuroprotective agents for Parkinson's disease in the lab, in experimental models, on animal cells, on animals, on in vitro models. Okay, great. So we have long list of brilliant minds and long list of ideas trying to solve the problem. Okay, try to go to the PubMed again and type the word neuroprotective agents for Parkinson's disease in humans. The list is going down. So the hundreds of thousands of papers are not transformed into application in clinics, are not transformed into trials in humans. This is the problem. Actually, the problem is increasing if you try to go further and try to search for clinical trials for anti-Parkinsonian treatment. Why? Because you might find one or two drugs that passed all the stages of clinical trial. So from moving from the initial phase of ideation, of working in the lab, until reaching the patient and reaching the community, usually the list of hundreds of thousands of research and ideas and thinking and teamwork is dropping down into one or two that might not be even successful enough to treat the disease. And this is a major problem. And this is why we are all scientists, all types of scientists, whether clinicians, basic scientists, geneticists, every type of those who are involved in biomedical research should start thinking in this challenging problem, which is how to cross the valley of this. Because as you can see, many of the nice ideas and innovations are falling in this valley and never regenerated again. Okay, so for that, many scientists, many visionists try to think, where is the problem? The problem is actually because we have two teams, we have two categories of thinkers. One of them PhDs or basic scientists, they are closing their labs and try to work on the fascinating world of genetics, signalings, molecules, and more. That's great. However, most of them do not have clue about the real needs of a clinician. So from a clinician perspective, for example, I'm keen to understand how to improve the quality of life of my patient, even if I'm not curing the disease. So for Parkinson's disease, clinicians, for neurologists, for example, it's okay, of course, if you are brilliant and came up with a real treatment that removes the disease, it's okay. But what if not? I shouldn't stop. My interest is actually improving the quality of life, improving the situation, even if I couldn't remove the pathology. However, from a biologist point of view, it's not worthy. So it's better to focus on the targets. This conflicts should be solved. Otherwise, we will continue to have two separate teams working independently in different directions. And this is a major problem. Do you think that translational research is worth funding? This was actually one question asked about 18 years ago in Australia. And the point of debate was, can we afford to fund translational research? And the lecturer actually concluded that we should move or change the title of the presentation into, can we afford not to fund translational research? And that's true. Now, with limited resource, every country has limited resource. Even the richest country in the world still have limited resource. Resources are not infinite. So with limited resources, we should focus on funding research that could have application, that could have benefits. Of course, every type of science is beneficial. However, we should always think of basic research with an eye kept on the future application. By funding translational research, I do not by any means ask funding organization not to fund basic research. No, this is not the concept. This is not the situation. I'm actually asking them to start funding basic research with one question: what would be your future plans? If your research pay off, what would be your next step? How we are going to move from your basic lab research into clinic, and then later on from the clinical trial into community engagement and acceptance of the innovation. With that said, I would like to share with you something called translational accomplishment. It is released by Society for Neuroscience, and actually it's a long time ago. This will highlight the importance of translational research in the area of neuroscience. Okay, this is the long list. Can you imagine? Several disorders and several diseases have been improved and had accomplishments only through translational approaches. One area is my area of interest, which, as you can see, Parkinson's disease. Can you imagine what is the translational approach that led to improvement in Parkinson's disease? It's not clinical trial. It's not working on patients. Actually, it is animal models. So development of better animal models for Parkinson's disease was a great step in better understanding the disease, on having a model for evaluation of neuroprotective agent, and even on understanding that critical pathways and the mechanisms leading to the disease. So better understanding, better evaluation models, and the more we improve the models or animal models for the disease, the more we have opportunities to advance research in this area. So as I said before, translational research does not mean working mainly on clinics, no, but working on basic science with an eye kept on the future applications. Okay, the first step we take in Egypt, and we have taken in Egypt a few years ago, was the establishment of Egyptian Network for Neurodegenerative Disease. And this was in 2013 as a collaboration between around 14 Egyptian universities, distributed geographically all over Egypt. And the aim was mainly to promote research in the area or in the field of neurodegenerative disease. And as you can see in the bottom, to further the dialogue between basic and clinical scientists. And I guess it is a model that should be adopted or generalized all over the MENA region. We have to adopt a model of a network that consists of basic scientists, geneticists, bioinformaticians as well as clinical scientists. Only through multidisciplinary teams we can appreciate and understand complex diseases or conditions like Parkinson's disease, dementia, and other neurodegenerative disease. Okay, so we started to think about what is the plan? What should be our goal, or what we call strategic or translational goal? And the answer actually was out there. So many publications worldwide were talking about what are the major challenges facing scientists in the battle against Parkinson's disease or other disorders. We started with Parkinson's disease because this is my area of expertise. And we had this answer from Nature Parkinson's Disease Outlook. As you can see, they identified 4 big questions. The first was, how does Parkinson's disease begin? And for that, they said that the next steps that are translational steps should be identification and defining novel biomarkers for early diagnosis of the disease. Great. What could be a next goal, another goal? What is the question was, what is the best way to divide people with disease into subtypes? Great. It's a great question. Why? Because actually the first step for precision medicine and precision health is to start stratifying patients. Now we should understand that each disease is actually consisting of different subtypes. So we don't have one Parkinson's disease. Actually, we have different types of Parkinson's disease. And only through genetic analysis and testing, we can identify several subtypes of the disease. So we can now identify a GBA Parkinson's disease, a LRRK2 Parkinson's disease, and more. However, the most important is to know and appreciate the fact that each population has their own genetic makeup. Each population have their own gene variants. Although we are all humans, we have different genes and gene backgrounds and risk genes. So we identify those two questions and 2 points as our ongoing plan for development of research in neurodegenerative disease in Egypt. And we started with some huge projects to tackle those issues. First was genetic studies. The first project, the first study was to understand the genetics of Parkinson's disease in Egypt. Again, we have to review few facts about the disease. We have 2 major categories regarding Parkinson's disease when we are talking about genetics. We have idiopathic Parkinson's disease. Idiopathic is the majority, constitutes the majority of patients where there is no familial history. So only one patient in the family with no signs of inheritance from parents or grandfathers. This is called idiopathic because we actually don't know the cause. We know by certainty that genes plays a role, however, not the only player. So in this model of the disease, we have genes, we have environment, we have this gene-environment interplay or interaction. That's why we started with this type of the disease or subtype of the disease, because it constitutes the majority of cases. We initiated the network of neurologists and scientists. We started adopting methodology, recruitment strategy, building a biobank, capacity building, and of course, recruiting funding. And the net result was 500 patients and matched controls. When you consider a project for more than 3 years with 16 centers all over Egypt trying to recruit patients, and the final results are 500 patients, you might say, okay, it's not that good. Yes, I agree. However, the value of the projects was the lessons we learned, not actually, or not only the patients, the lessons we learned, the mistakes we made that helped us to learn how to build our network, how to improve our ongoing research, how not to fall in the same mistakes in the next project. In addition, I guess the output was quite good. We could identify new risk gene for Parkinson's disease. We could identify new risk factors, environmental factors for Parkinson's disease. And more important, we identified an area of interaction between those environmental factors and genetics. And the end result was this paper. I guess the more important result, however, was that we found that most of the well-known risk genes for Parkinson's disease are not out there in Egypt. We don't have those genes. So we screened around 80 single nucleotide polymorphisms, as risk genes for Parkinson's disease, and we got only one, and one which has not been mentioned before for Parkinson's disease. So this was a very nice lesson that our genetic makeup is totally different. There are some changes, there are some differences. We still have to do our own homework to understand genetics of disease and genetics of Egyptian and might be North African populations. That's why we continued for next step. We are start confirming the data on functional levels, cell levels. We are now studying the other environmental aspects like lifestyle and diet. And more, in addition, we started to understand the genetic background of the other subtype of the disease, which is the familial or genetic type. Familial or genetic type of Parkinson's disease constitutes the minority. They account for around 10 to 15% of cases. However, although they are a minority, they pose a very strong point, scientific point, which is they help us identify genes with certainty. For idiopathic Parkinson's disease, you identify certain risk genes. So this means that those genes could increase the vulnerability to the disease, but you are not certain 100% because you have different confounders. However, in genetic or familial types of the disease, you can identify single mutation in single gene that could lead to the inheritance of the disease with simple Mendelian inheritance model. And this is very important actually to understand the tracks and pathways that leads to the disease. So it is, in addition to identifying new genes for the disease or identification of genes causing the disease, you could also identify the tracks and the pathogenic targets for treatment and for biomarkers for the disease. So we started this project as well. And as you can see, the first initial result was identification of this rare mutation in an Egyptian family. However, we continue now to assemble an Egyptian panel for Parkinson's disease in familial cases. It's not the end of the day. So you might notice that usually you start with one step. However, next steps are not predicted, not expected. So based on our previous studies, we have been working for around 6 years or more on genetics of Parkinson's disease. We discovered a very important point that our genetics, as I said, is not similar to European ancestors or American genetics. That's why we cannot depend on the reference genome, which is available, the human reference genome based on European ancestry, on our analysis of the data. And here comes a recent output from our network, which is assembly of the first Egyptian reference genome. And by reference genome, I mean genome for normal population, healthy population. In order to identify potential pathogenic mutations or SNPs or variants in genes, we need first to know what are the normal variant in Egyptian population. And for this project, we started since 2016, and the analysis took us around 3 years to start a de novo assembly. De novo means from the scratch reference genome for Egyptians, comparable quality to human reference genome, and with comparable quality to other reference population genome like Korean, like Chinese, like other population genetics reference genomes. And I guess it is the first North African reference genome so far. Hopefully it will not be the last. So as you can see, ENND started with simple study or project for analysis of risk or identification of risk genes for Parkinson's disease, and ended up with drafting of the first Egyptian reference genome. This was the first track, and I guess we were doing very well in this part. Currently, ENND is collaborating with IBDGC, joining IBDGC Africa to contribute more and understand more about genetics of Parkinson's disease in the whole African continent, so through comparison between different genetics and risk factors of Parkinson's disease in Africa, not only Egypt, but all over Africa. Okay. The other aspect or the other translational goal we were building was biomarkers. And biomarkers are very important when we are talking about neurodegenerative disease. Why? Because simply when you start clinical manifestation, when you start complaining of Parkinson's disease, when you start feeling those tremors and bradykinesia and instability, and seeking clinical care or medical consultation, this means that actually you lost around 85% of your neurons. So the treatment options are very limited. The window of improvement is very narrow. You don't have room for improvement or neuroprotection. Here comes the importance of biomarkers in neurodegenerative disease, because if you can predict the disease before clinical manifestation, if you can predict the disease even before the loss of 50% of your neurons, then might be there is a hope. That's why most of research areas in this field is focusing on developing novel biomarkers. But by novel biomarkers, especially in areas like Middle East, I would say that we need accessible, low cost, easily done, performed biomarkers. For that, we started trying to validate and come up with sera-based biomarkers in patients of Parkinson and Alzheimer's disease. We adopted not a new methodology, but we try to improve, searching for autoantibodies against cytoskeletal proteins, against neuronal cytoskeletal protein. So usually in case of brain damage or neuronal damage, the proteins inside the neurons are escaping, going to the sera or the systemic circulations. The immune system start to be triggered, releasing antibodies against those proteins. We are detecting those antibodies. So we started with 2 or 3 autoantibodies, and then later on, we increased the panel to around 10 autoantibodies against neuronal cytoskeletal protein. And our hope or our aim is to reach to specific pattern for each disease. So hopefully, we hope that at certain time, we could say that the pattern of elevation and depression on those 10 autoantibodies battery could help identify Parkinson's disease or predict Alzheimer's disease. Could say that this person is in risk of developing Parkinson's disease within 10 years, for example. It would be a groundbreaking discovery. However, when we started thinking about biomarker, it seems appealing to know that it is not only, and here comes the area of translation. It's not only biochemical biomarker. Sometimes it is a biochemical, sometimes they are genetic biomarkers. Sometimes even we could have crazy or innovative ideas. So this is the latest research we have been involved in the ENND, and it is funded by Alzheimer's Association and the Alzheimer's Society, as well as Global Brain Health Institute. So we are trying also to pick up the changes on your interaction on Facebook. We feel that it's better to develop a system, a paradigm for early diagnosis rather than depending on one system. So now we are working on interaction on Facebook and social media, biochemical autoantibodies, and even neuroimaging using artificial intelligence. I think that in a few years, engaging or incorporating different input from different systems, social media, early clinical biomarker, biochemical biomarkers, and even neuroimaging techniques, we could adopt or come up with a new innovative paradigm to early diagnose a disease. However, we have one major challenge, is that we are trying all our biomarkers on patients already diagnosed. So we are not solving the problem. We might say they are diagnostic biomarker, but we need predictive biomarker. So to start validating your results or your innovation or your discovery, you need a longitudinal study. That's why Egyptian Network of Neurodegenerative Disease now is launching a longitudinal study of Egyptian healthy aging. The main aim is to have or to recruit a number of patients and follow up them through 10 years period. So can you imagine you could draw blood samples and start doing your test and come up after 10 years and say, okay, based on our initial result, we expect that subject 1, 2, and 5 should develop Alzheimer's. And you find that you are right, or maybe wrong. But this is the only way for validation of your biomarker studies. And we started the launch of the pilot in collaboration with SHARE, which is the longitudinal study of European Union. We got funding from the DAAD, German Academic Exchange Services. And now we are one of the registered HRS harmonized studies worldwide. I hope that I was not boring. I hope that I didn't take a long time. I just would like to end with this quote. It's always impossible until it is done. So Don't believe there is impossibles. There is always possible impossibles. Thank you so much.